Good evening, ma'am.
Good evening, sir.
Good evening.
Sir, you can start now, sir.
Yes, ma'am. Thank you. Thank you so much. Hi, and welcome to the wonderful session of the biological classes, my dear students. You are all welcome. Today, what we are going to discuss about, bioremediations. It is nothing but the part of biotechnology and application of the biotechnology already in our about. Bioremediations. Bio means plant as well as animals already in our but remedy. It's nothing but the what is that? How it will be recover the environmental damages with the help of the living organism is called as bioremediation. The application of the biotic agents, but what is by biotic agents? Biotic is nothing but a non-living substance, so such as light, temperature, and pH, and water, air, and all those things comes under the getting known as biotic or abiotic. Abiotic. Okay. Biotic means the living organisms. What is the living organism? All the living organisms, whether it may be lower organism to higher organism, prokaryote to eukaryote, they are all comes under the getting known as biotic agents. Okay. So what are the biotic agents like? Microorganisms in the correction and the recovery of environmental damages is called as bioremediations. Bioremediation, living organism used for the correction and recovery of environmental dangers is called as bioremediations. One of the best examples in this part, one of the best example is Pseudomonas putida is nothing but the one of the super bug. It is also one of the microorganisms. It is used for the removal of oil spilled on the sea water by help of bacteria is one example. How it will be already discussed in yesterday class, isn't it? You know what about the petrochemical is made up of otherwise called as crude oil. The crude oil is made up of complex hydrocarbon. Hydrocarbon, you know very well. So it is made up of carbon, hydrogen, oxygen, isn't it? It is nothing but the more than one polyhydrocarbons united together form as a petrol or ether or tar, kerosene, okay, and phenol components, naphthalene, and so on. So we are getting a total 10 to 20 fuels. We can easily obtain different fractionation, distillation process. The crude oil is complex hydrocarbon, and how it will be, that complex hydrocarbon split on the ozone layer. So as a result, what happened, the aquatic animals are well affected. It cannot able to breathe itself. Not only that, and also the light also, it will not penetrate into the water molecules and water bed. So as a result, the aquatic animals also, they will not get proper light. So at the same time, some of the plants, okay, they will not get the light. They will not do their photosynthesis reaction also. So the aquatic animal as well as the aquatic plants also suffered. That time what they will do, the one of the super bug, Pseudomonas putida, it is also spray on that surface of the ozone. Okay, the Pseudomonas putida, what they will do, they act upon the complex hydrocarbon. Okay, it will be split into simple compounds such as carbon, hydrogen, oxygen, nitrogen, sulfur, respectively. As a result, waterbed completely, what is that? Recover into the, it is also turned into the normal sea. So that is very, very important, my dear students. See that bioremedy. Okay, these are all are already in our world, but there are so many chemicals also exhaust from the laboratory. Sorry, laboratory of the any one of the industries, isn't it? Not only that, all the microorganisms, take an example, carbon monoxide in the form of smoke, and also it exhausts some contaminated water. It is also affect the land or water bed. Okay, seashore, whatever it may be. They eliminate the carbon dioxide. See that carbon dioxide is also. It will be, what is that? Treatment with the microorganism, that is called as bioremedy. Already but one of the best examples, sewage treatment. Okay, and sewage treatment is nothing but about there are three techniques. Is there one? First primary treatment, secondary treatment, tertiary treatment, and so on, so that. Okay, as a result, what happened? The contaminants and the impurities completely removed from that particular molecules, and also it will be converted into a useful form, impure to pure form, and also used for the drinking purpose or irrigation purpose. Likewise, after that, that carbon dioxide is also recovered by the plant, it will grow, and also it converted into carbon dioxide into useful products such as starch and some other forms. Already about that things, isn't it, my dear students? Don't forget, this is the important things. Next, the bioremediation is also divided into two types according to their nature. One is called as in situ bioremediation, and another one is called as ex situ bioremediations. Further, in situ remediation is also divided into two: intrinsic bioremediation, engineered bioremediation. Okay, the engineered bioremediation is also divided. So many subdivisions is there. See that biosparging, bioventing, bioslurping, biostimulation, bioaugmentation, natural attenuations. So these are the types also comes under the engineered bioremediations. Exit bioremediation also divided into two parts. It's slurry phase bioremediation and solid phase bioremediations. The slurry phase, you know, slurry is nothing but the semi-solid nature. Solid already in our about. So here, what is that? Bioreactive act as a slurry phase bioremediation. Already we discussed it. Bioreactor. Okay, there are three stages also there. Each one it has a specific rotator. Okay, and solid phase bioremediations, biopilling, land forming, composting, and also a final one is called as biofertilizers. These are all comes under the headings known as bioremediation. Okay, my dears. So here, in situ, ex situ. So what are the intrinsic, engineered, slurry phase, and also solid phase? Here, what are the types? Each one it has a specific functions. Each one it has a specific components. Okay, properties also that. Okay, so that one no need. But here you must note it on bioremediation is divided into two. It's ex situ, it's outside, in situ within the things. Land forming, biopills, bioreactor, composition, it's also ex situ. In situ, natural attenuations, bioventing, bioslurping, biosparging, these are all in situ. Sometimes they may ask the questions and match the following: bioremediation ex situ, which one is not comes under the heading, or in situ, which one is the related to the in situ bioremediation, like that they may ask the questions. Okay, don't forget, my dear. Next, we will pass on to the next one is called as gene therapy. It is also one of the bioremediations, my dear students. Gene therapy is a collection of methods that allow correction of gene defect diagnosed in the child embryo. Very, very important. Take an example, say any one of the family. Their generation, it contains so many defects, that is called as gene defect. How will you recover these types? Already we discussed it. You have to identify the particular gene responsible for the any disorder or defect, gene defect. What are the gene defects? Already we studied in chromosomal aberration, and also X-linked genes resistance, X-linked genes. What is that? Disease. You know, phenylketonuria, hemophilia, and also alkaptonuria, and also color blindness, and so on. So there, isn't it? These are all how will you recover? With the help of the gene therapy, we can control all those things. Okay, we can control it. How? In the embryo stage, you have to identify the embryo stage. The gene is, you have to isolate from the DNA, identify the particular defective DNA is also isolated from the DNA molecule. Then already know the interesting gene is also inserted with the help of the biotechnology and genetic engineering. And again, after that, you know about the morula state. The cells also undergo the cell division process. As a result of formation now, morula stage or blastula stage. After the blastula stage, it will be replaced into the mother. Okay, that process also done. So in that case, the result will be in good condition. That is very, very important, my dear students. This is important. The correction of genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. This was first in 1990 FDA. Okay, FDA for the first time approved a gene therapy experiment on ADA-SZID. What is my ADA? Okay, in the United States, after that treatment of Asante D. Silva. Okay, this was first gene therapy was done by which one? The name of the person, Asante D. Silva. Don't forget which year, 1990 FDA. Okay, which experiment they did, ADA, here FDA, here ADA, SCID. Okay, see that one. What is meant by SCID? And basics of gene therapy. Normally, what are the basic principles they are following? First one is replacing the defective gene. That's already I told you. You have to isolate the defective gene by the deletion method. Already in our chromosomal deletion or gene deletion, already whatever it may be. Then you are going to insert the normal gene. So that is the first point. Addition and deletion, or deletion and addition, whatever it may be. And then repairing the defective gene using appropriate method. This is the method. What is the method we are using here? Biotechnology, genetic engineering. Okay. That is called as what is the fusions. Okay. What is the fusions? Already know a little bit. Two genes also involved in this process is A strain and B strain. A is responsible for the defective one. B is there responsible for the superficial characters, like that. You will cut the gene exactly the particular genetic material. You cut the gene with the help of the restriction enzyme and replaced by the super gene or and what is the interested genes also, that is also inserted with the help of the DNA ligase. So after that, you will be transferred to the host cell. Within the course, already know there is a normal process. Already we discussed so many things. And the third one is modifying the regulatory mechanism or the operon of the gene so as to gain control over its protections. It is nothing but the regulatory genes. Regulatory genes, what does that mean? Switch on and off. Suppose when they need, what happens automatically? Genes also activate in on conditions. That time what happen? What are the process? It will be reduced. Suppose the production will be more or high, that time automatically what gene is also undergoes the process, gene expression in off conditions. That time what happened, the process will be completely stopped. Stop the productions. Already we know about that things also in lac operon concept, isn't it? So that is the important point. And types of gene therapy. So what are the two types of gene therapy? The one is called a germline gene therapy. And second one is somatic gene therapy. There are two types of gene therapy have been reported. Okay, see, one is called as germline gene therapy. Germs, that is related to the, what is that? Sperm or egg cell. Here the gene therapy is applied to germ cells. Germs means what is that? Sperm or egg, whether it be a male gamete or female gamete. Animals. Okay, don't forget. The somatic gene therapy. Here the gene therapy is applied to the somatic cells. The somatic cell is nothing but the body cells. Don't forget. Here the reproductive cells. Here the somatic cells. Don't forget, my dear students, germline gene therapy is mostly used for which cells? They may ask the questions. Germ cells. Germ cells, otherwise called as reproductive cells, such as sperms and the egg. Okay. And somatic gene therapy. Somatic is nothing but what is that? Body cell. It occurs only in the somatic nucleus. Here the germ nucleus. Don't forget, my dear students. Okay, go to the next one. So mode of gene therapies is the germline and somatic. The germ cells are modified by the introduction of the functional genes, which are integrated into their genome. Okay, banned at present. These things is completely banned. Okay. The somatic gene therapy, genes are transferred into the somatic cell of a patient. This is not banned. So this is the banned one because so during this process we can create our own, what is that? Pictures. I mean, own structures. Okay. So that is an important one. See the gene therapy. Here, cell with a non-functioning gene. Here, there is no cell, but non-functioning. Here, the functioning gene is there. Okay, the functioning gene is introduced into the cell with non-functioning cell having, but non-functioning gene is there. So now, what happened? These two are compound contact close together with the help of gene therapy technology. We are introducing into the functional gene into the non-functional cell. So as a result, it present, what is that? Cell function is normal. Okay. And then another one is called as, what is that? A gene inhibitory therapy. Sometimes the production is over. What happened? They are using the blocking genes. Blocking gene is also responsible for the controlling of the, which one, functional genes. As a result, what happened? The new gene product blocks cell function normally. What is that? Affected. So these are the two steps. When they need, they will use. When they will not need, they are using the blocking genes. As a result, what happened? The product will be completely stopped. This is called a gene regulation. Already we studied the lack of concept by Pidil Tratum. Okay. Jacob and Monod. Okay, don't forget. See, then gene therapy can be used at two different levels, my dear students. So what are the two different levels? One is called as embryo therapy, another one is called as patient therapy. Already you know very about what is meant by embryo. What is meant by embryo, my dear? You know very about. Embryo is nothing but the fusion of gamete. After the fusion of gamete, what happened? It will be converted into zygote. Zygote develop into, what is that? Embryo. That is in which the genetic constitution of the embryo at the post-zygotic level is changed so that the inheritance also changes. This has been tried in case of mouse and rabbit only. Okay. The embryo therapy is also done. Which level, like that they may ask the question, it's the post-zygotic level. That is very, very important, my dear student. What are the animals they tried? There are two animals have been reported. One is called as mouse, another one is called as rabbit. Embryo therapy. Second one is patient therapy, in which cells with healthy gene may be introduced into the affected tissues so that the healthy gene overcomes and defect without affecting the inheritance of the patient. Already know about these things is nothing but the stem cells. When you are introducing the stem cell, any part of the body, what will happen? The particular part will be grow, or particular organ it will be grow. How? In healthy conditions. So these are the two things have been reported, my dear. Okay. So don't forget, my dear students. Next one is, for this first, the defective genes should be identified. First of all, you know about which one is the defective gene. You should identify the defective genes, and then we can replace the defective gene into normal healthy gene. Then second one is, normal healthy genes should either be isolated or synthesized. It may be isolated, means any one of the organisms, a living organism, we can isolate. Synthesis means artificially synthesized in laboratory. And then cell of the tissue where the normal healthy gene will need to function should be placed into a cell where it can function. For example, take an examples. Okay. The bone cell is there. Take an example. Okay, my dear student. This is very, very important, my dear. Don't forget. Okay. So take an examples of. Okay, take an examples. So this is the considered here. Okay. So this is considered a bone cell. Okay, bone. Here there is a break. Okay. Here there is a break like this. Okay. In this part. There is a small break is there. So I will come to the normal one, so then it will be better. Okay, see here. The bone is there. Okay. So this is the normal one. Okay. Okay, the fracture is here taken. Okay. Just imagine. So normal bone, it will be, how is it now? So this is the normal bone, isn't it? This is the normal bone. this bone is how it will be. So there is some breakage. Okay, some breakage is like this. It's not like this. Suppose it is like this. Okay, there is a small breakage. Okay, how will you recover this one? You can plate. You have to place a plate, isn't it? There is a plate. You can use it some remedy. What is that? How it will be? You can use it any one of the plate like this in both side. They will place their plate. Okay. Suppose it is small changes, small damage is there, small damage like this, small damage like this in this part only. Okay, that time what they will, they will use the stem cells. They will use the stem cell. This is considered a stem cell. Okay, okay, stem cell, stem cell. Okay. When you are using the stem cell, what happened? No, stem cell where it will be isolated already in our body. Stem cell is also isolated from the umbilical cord during the delivery. Okay, so you will get the, what is that? So you will get the, so it is also connected with the fetus, isn't it? It is also connected with the fetus. Okay, so it is also connected with the fetus. The umbilical cord is there. Here, the what is that? No, stem cell is there. Stem cell, it is identified. Stem cell. What is the main use of the stem cell? It is used for the future. It is used for the future. It's a baby, isn't it? Baby, always called as during the month. What is that? Delivery time. What happened? No, you can get the stem cells. Okay, the stem cell is placed here. Okay, allograft, autograft, already know, but there are so many methods is there. Introduced means the bone completely, what happened, developed. Okay, the bone is completely molded with the help of the stem cell. It produces the new cells. It is also one of the gene therapy. Okay, so some cases stem cell bank is also there. Already we studied in. Okay, these are all one of the gene therapy methodology, my dear students. Don't forget. Okay, here you must note not only here and also kidney also. Kidney. Take an example of any one of the kidney. Okay, so kidney cells also. They are having some, what is that? Here. So some, suppose if it is any damage in the end part, nerve cells also, nerve cells. Okay, nerve cells. You know about the nerve cells look like this one. Okay, nerve cells. Some of the nerves also affected by this one. It will be recovered by the gene therapy. It is also important, my dear students. Don't forget. Okay, go to the next one. Okay, okay, my dear students. Don't forget here. And next one is in vivo gene therapy. What is meant by in vivo? In vivo, in vitro. In vivo method of gene transfer involves the transfer of cloned gene directly into the tissue of the patient. This is the important part. In vivo. Okay, directly into the tissue of the patient. Okay, which one? The gene transfer. Gene transfer through the method is called as clone gene directly into the tissue of the patient. This is done in case of the tissue whose individual cell cannot be cultured in vitro in sufficient numbers like brain cells, and or where the re-implantation of the culture cell in the patient is not efficient. Suppose some of the person suffered by any one of the disease, that person that person is from the patients, we can collect the sample or cells or genes. Okay, it can be cultured. Okay, culture. It will be re-implanted the particular person means, suppose if it is accepted his body, it's okay. Suppose if it is not accepted by his body, what happen, it will lead to some difficulties. Okay, that condition, what happening, they will introduce directly into the tissues, in vitro conditions only. That is very, very important, my dear student. This is done in the case of tissue whose individual cells cannot be cultured. Suppose some of the percent cells, they cannot be able to culture in the laboratory aspects, such conditions. Okay, and also already about like a brain cell and or where reimplantation of the culture cell in the patient is not efficient. Suppose once you culture the patient cell in laboratory aspects, it is not efficient. It is not the ability to function all the activities. In that condition also we are using the in vivo gene therapy. Okay, my dears. So go to the next one. See that in vivo gene therapy. So this is the person. We are selecting the genes. So we are collecting the cells. Cell inside there is a nucleus. The nucleus is analyzed, isn't it? Analyze which one is we can remove it, whatever it may be, and also culture it. Again, reload. We have to introduce into the person. Suppose this gene is accepted by his body. It's okay. Sometimes it may be not efficient like as a human cells. That is also another one. Ex vivo therapy. It is another type, in vivo and ex vivo. In this method of gene therapy, genes are transferred to the cell grown in culture. There, what they will did collection of sample from the collection of gene from the particular person, and also some changes occurs, reintroduced into the same person, isn't it? That is called as in vivo. Ex vivo means, what is that meaning? Collection of sample, collection of gene, and also a culture it. After the culture finish it, and then re-enter into the same process, same person. That is called as ex vivo therapy. Ex vivo, in vivo, there is some difference is that ex vivo is cultured one. See that in this method, the gene therapy genes are transferred to the cell, grow in a culture. After the culture gene cells also transfer to the selected one. That is very, very important, my dear. Transformed cells are selected, multiplied, and then introduced into the patient. That is very, very important. Don't forget, my dear. Ex vivo is nothing but the cultured gene introduced into the patient. Okay. First of all, you should select the person and collect the sample, and also multiply and then reintroduce into the same person. It is called as ex vivo. But there, a collection of sample, you did some physiological changes and reintroduce into the same patient. That is called as in vitro. Got it? See that one. So this is the in vivo gene therapy. This is the ex vivo gene therapy. In vivo gene therapy, what they will do, see that samples collected from the persons. And also, so non-viral delivery system, the viral delivery system. These are the two things done by this one, in vivo gene therapy. Okay. Here, ex vivo gene therapy. What is the meaning of ex vivo gene therapy? Okay. Ex vivo gene therapy is nothing but what is the selection of cell isolated from the patient. Here, cell isolated. The cell is cultured. Then culture is also modified, some forms, okay, wanted and unwanted characters removed, and also addition of the superficial characters. After that, it will be introduced into the same person. This condition is called as ex vivo gene therapy. Don't forget, my dear student, this is very, very important. Ex situ and in vivo. Ex vivo, in vivo. In vivo means you are taking the cells and also directly introduce. The ex vivo is nothing but the culture, the isolated cells are culture and then reintroduce. That is very, very important, my dear. Don't forget, this is the important things. Same things is also in your book. Okay, my dear. So go to the next one. So in vivo and the ex vivo. Okay, in vivo means therapeutic genes. Okay, so we are selecting And again, we are introducing how it will be from the any one of the person suffered by the viral disease. It is the disease fight against many diseases, means it will produce some component inside, isn't it? You have to identify, directly inject into the person. Here, take a identification of the person. Person is nothing but the diseased person, collection of stem cells, and culture it, and modify it, and also, what is that? Introduce the therapeutic gene is also here. After that, modification of this one is also transferred to the same person. This is called as ex vivo. Okay, so that is very, very important. So what are the technique they were used, my dear students? Just note it down. This is very, very important, my dear. What are the technique involved in the gene therapy? The isolation and analysis of gene is gene of interest. Which gene is we want to introduce into, or correct it. We want color blindness, cancer cells, phenylketonuria, hemophilia, or whatever it may be. What are the diseases already, will be gene related one. It can be isolated in the analysis of gene of interest. Okay, somehow the person, it has a, what is a hair in the white color, isn't it? In North, in other country people, European peoples or some other peoples, their color will be white in color. Okay, if you want to change the color of that person, you have to isolate the white hair color gene. Replace means, will be black color, isn't it? Already but that things also we are having black. Some of them are having brown in color. Okay. Likewise, the eye colors also change. Black, blue, and also that blue is color. Blue is green in color. Okay. So like that, it is also responsible for the particular gene. If we want to change, means we are using some, and take an example, so many actors, they were used, what is that? The eyelids and the lens is also changed, isn't it, for their commercial purpose, isn't it? But naturally, if we want to change, means what happened, we can identify the gene interest. That gene you have to isolate and analyze, and also the technique is also used, then you will get the normal person to that blue color eyes or some other superior quality you will obtain. Second one is construction of the normal gene. It is also very, very important. These are the gene therapy, construction of the normal gene. And third one is transport of gene construct into host cell. Transport of gene construct into host cell. A little bit after the construction, suppose you are creating your own gene. For example, you are having, if you want to fly, you need wings, isn't it? That gene isolated. Okay. If you want to run like a cheetah, okay, you have to use you have to identify the particular gene for running process. Okay. And also jumping from here to there. Take an examples of kangaroo. Each genes you have to isolate and construct your own gene and also reintroduce into the host cell. You will get the 3 characters. Okay. After the obtaining of 3 character gene, it will be introduced into the human being. So that person, it will be jump and hair, and whatever it may be, all the characters will be expressed. And fourth one is integration of the therapeutic gene into the host cell. Therapeutic is nothing but the medicinal oriented. You know all about that. Tetracycline resistance, ampicillin resistance, and so, isn't it? That things also. And then vector, otherwise called as carrier molecule, must be used to deliver the therapeutic gene to the patient's target cell. The vector is also very, very important. Without vector, there is no any genetic engineering process. It will not occur. That is very, very important, my dear students. Okay, already humulin already but it is nothing but a human insulin produced by laboratory with the help of the vector is called as E. coli, Escherichia coli, otherwise called as E. coli. It's a vector. Okay, see good. These are the important. The most common vector is virus that has been genetically altered to carry normal human DNA. You know about that virus, it is the pathogenic, isn't it? Suppose the gene is completely removed, the disease producing gene is removed, And also sometimes it may be altered, genetically altered to carry the normal human DNA. So the particular virus, what happened? It will not affect any disease. It will not produce any diseases. Okay, it will not produce any harmful effect because already we changed it, isn't it? It's a one. A vector should be safe, efficient, and selective. It is also very, very important. We cannot be able to introduce any vectors. We cannot select any vectors because which one is easy, which one is handled easy, which one is give a good result. That things you should be select. Suppose take an example. Any one of the COVID is virus is already they told, isn't it? Virus is also one of the vector, isn't it? We will not choose the COVID virus. We will not choose the AIDS or carcinogenic effect producing viruses because it is very, very important. Okay, safety is the first measure. So that's why they have told vector should be safe, efficient, and also selective. And then gene therapy was first applied in the treating a 4-year-old girl. This is also asked. This question is also asked so many times, my dear students. Asanti D. Silva suffering from an immune system deficiency in the year 1990. Don't forget, my dear students. Who suffered? Who was first treated by the, what is the gene therapy? Which girl, which name of the girl? Like that, they may ask the question. Asanti D. Silva. Is that name? Asanti D. Silva. Suffering from an immune system deficiency. Sometimes they may ask the question. The Asanti D. Silva suffering from which type of disease? Immune system or any other? Color blindness. Okay. And hemophilia. Like that, they may ask the questions. Okay, the treatment is also occurs which year, like that they may ask the question. 1990 gene therapy treatment was given by that particular girl. Which year? See that 4-year girl. This question is also asked. How many years that girl age is? How old are you? That girl, like that they may ask the questions. 4-year-old girls. Asanti D. Silva suffering from an immune system deficiency in the year 1990 in the treatment of S-linked server combined immunodeficiency. Here, S-linked. Okay, S-linked is denoted sexual chromosome linked. That's why it is given as X-linked. See, SCID. Don't forget, my dear. SCID. A server combined immunodeficiency. SCID. X-linked. Don't forget. Okay. So these are the important points. In this point, there are 4 questions. Is the first one, name of the girl. Next one, which disease. Third one, which year-old girl. Fourth one, which year. Fifth one, the treatment given by the method is called as S-linked server combining immunodeficiency. SCID. Don't forget, my dear. This is very, very important. The key immune system cell called as T cells were not working because they lacked the enzyme ADA. That conditions what happened, the particular girl suffered by a immune system, isn't it? The immune system, what happened? No, but immune system is responsible. Which one? There are 2 cells is there. T cell and B cell. Both are producing lymphocytes, isn't it? Lymphocytes fight against many diseases already know about. But here, the T cell is responsible for the, it's not working. Why the T cell work? If we want to work the T cell means they need some enzyme. That enzyme is called as ADA, adenosine deaminase. Don't forget, my dear. This question is very, very important. So many times they were asked the question, which enzyme responsible for immunodeficiency? Okay, ADA, adenosine deaminase. Which cell? T cell. Which enzyme? Adenosine deaminase enzyme. Responsible for the immune system. That is very, very. Without these cells, and so she could not mount immune response against invading pathogen. That girl suffered. What is that? Due to the T cell were not working due to the lack of enzyme adenosine deaminase, otherwise called as ADA. What happened? It will not fight against diseases. So she will fall on sick very shortly. Very shortly. That is the reason. Without these cells, and so she couldn't mount immune system. Immune response, again, invading pathogen. Pathogen is nothing but the disease-producing microorganism, any other foreign particle. This enzyme is crucial for it, immune system to function. So that is very, very important. So that gene is isolate and also produce the ADA genes. See, the disorder is caused due to the deletion of the gene for adenosine deaminase in 25% of these cases. Those who are suffering from these diseases, what happened, the particular T cell producing amino enzymes, that is adenosine deaminase gene, producing gene, 25% is, what is that? Deleted. Only 75% is there. So we come to the conclusion, 100% should be there. Adenosine deaminase production gene should be there, then only it will be fight against many diseases. So it will reduce means what happen, the production of the amino enzymes also, what happen, reduce. As a result, microorganisms or easily affect the person, just like AIDS persons. These patients have no function T lymphocytes. They already told you. So T lymphocytes is essential for the disease. In some children, ADA deficiency can be cured by bone marrow transplantations. How it will be transferred, So you know what about the bone marrow transplantation. Bone marrow means already you know about that things also already we discussed last. See this one. This one. Just imagine. Okay. Okay, bone marrow. Already you know all about what is meant by bone marrow. You know all about. So this is considered. This is considered bone, isn't it? Okay, this is considered bone. Okay. Okay. Inside there is a solid substance present here. Okay. Inside there is a solid substance system. This is called as what is that? Bone marrow, isn't it? Bone marrow. What is the main functions of bone marrow? You know very well. The bone marrow is very, very essential for the synthesis of blood component, whatever it may be, blood. Okay, blood. Okay, blood component. What are the component? Okay, blood component. Okay, blood components. So what are the blood components you know about? Okay, already know about. Okay, so RBC, very, very important. RBC, isn't it? Immune response, okay, WBC. And also platelets. Okay, platelets. Okay, platelets as well as RBC both are involved in the blood clotting mechanism. WBC is fight against many diseases. WBC because it contains nucleus, isn't it? So here the suffering person of the baby, the bone marrow transplantation occurs. What happened? The healthy bone marrow donor, healthy. Okay, very, very important, my dears. Healthy bone marrow. Okay, healthy. Okay, so. Okay, see that this part completely remove from the donor. So the bone marrow is completely removed. Okay, this is the donor. This is the donor. Okay, here the donor is there. So the removed the bone marrow. So this is considered bone marrow. Already you know about this is the bone marrow. Okay, so the brown color, red is brown in color card part. It produces the RBC, WBC, erythropoiesis, leukopoiesis, platelets, and so on, isn't it? So it can be transferred to the affected baby bone. So affected baby bone means same process you have to do. You have to cut the bone, isn't it? Cut the bone, and inside what they are having, you can choose another this bone marrow. Okay, so from you have to collect the bone. Okay. Okay, this is the patient, isn't it? So here you have to replace into healthy donor to the person. Okay, replaced here. Okay, here replaced. So this is the bone marrow. Okay. So now what happened, that's a healthy bone marrow is also inserted into the where, unhealthy or disease containing patient, isn't it? Disease producing patient, isn't it? So now what happened? These bone marrow produce the healthy, what is that? WBC, RBC, and platelets. So this is the important, my dear student. This method is used for the gene therapy. Okay. As a result, what happened? The person, it will be live for some more days. That is very, very important. Some more days. That is very, very important. Okay. They can survive few days more than that. That is very, very important. So, okay. Say that. So these patients have no functions of T lymphocytes. In some children's ADA deficiency can be cured by the bone marrow transplantations. In other, it can be treated by enzyme replacement therapy. Enzyme replacement, because the adenosine deaminase in enzyme, isn't it? It not produced means what happened? It can be replaced by enzyme replacement therapy, in which functional ADA is given to the patient by injections, just like insulin. They will give ADA intravenously. So it acts as a T lymphocyte production. So as a result, what happened? The person is escape from the pathogen. That is very, very important, my dear student. Gene therapy has been found successful in treating several genetic disorders, including SCID, thalassemia, cystic fibrosis, muscular dystrophy, and certain kinds of cancer. Already knowable. Blood cancer is also there. Blood cancer means, what is that? The particular blood cell is increased in the particular lymphocytes or monocyte like it. These two number will be 100 million fold present in the blood cancer percent. Okay. Normally, but 1 drop of blood in normal person, it contain lymphocytes 65%, in those who are suffering the blood cancer, that you collect the sample, you can analyze it. 1 drop of the blood, it contains 65,000 of monocytes, 65,000 of lymphocytes, like that, That is the divisions of uncontrolled divisions of cells. Already the cancer is nothing but the uncontrolled cell division is called as cancer, isn't it? That is the patients. It can be replaced. It can be curable by the method is called as gene therapy, particularly bone marrow transplantations. Like cancer, which one? The AIDS patients also. AIDS patients also lose their, what is that? Immunity powers, isn't it? That time what they will do now? Bone marrow transplantations also done. So as a result, the person, it will be long. It will be possible to live for some more days. That is very, very important, my dear. Okay, don't forget. Next, procedure for gene therapy. There is some special procedure is that you have to follow it. Then only it will be recover. Otherwise, it will be very difficult. Dr. Michael Blasey removed the genome lymphocytes from a bone marrow which had defective gene and cultured them. Okay, he did what they did. He removed the genome of lymphocyte. Genome is nothing but from the lymphocyte, the gene is isolated. That is, DNA molecule is isolated, and culture them. To the cell culture, they then added bioengineered viruses. That is retroviral vector, into which they had incorporated working copies of the ADA genes. Already, once the virus enter, what will they do, my dear? Virus enter means virus gene is also incorporated with the host gene, isn't it? The multiplication takes place. Likewise, what he did, the specific lymphocyte producing genes are isolated and cultured and replaced into the virus vector. So now what they did, what what will happen inside, the virus gene carry the lymphocyte producing gene, isn't it? So it will multiply inside, where lymphocytes production is more, isn't it? Like that. See that? Incorporated working copies of the ADA genes. The ADA genes within the virus. When the virus infected the blood cells, they inserted the ADA genes into the cells. The treated cells were then re-injected to the patient bone marrow. So now what happened, see here, what he did, defective lymphocyte genes also isolated and culture. After the culture, he select the retroviral vector. Okay, retroviral vectors, what happened? The ADA gene is inserted into the virus. As a result, what happened? The virus containing ADA gene. This gene is reinserted into the bone marrow. So what happened? The bone marrow containing the nutritive substances, it will be absorbed by the virus. It will produce more amount of ADA gene, isn't it? That is the important idea. See here, the lymphocytes subsequently produced by these stem cells had a fully functional ADA genes. Already told you. After that, T cell expressed the ADA gene, and the cellular and humoral immunity was restored. What they did, there is a special procedure is there. First of all, lymphocyte is very, very essential for the functioning of the T cell. The lymphocyte and T cells activity was done by ADA. So you have to find out the ADA gene. The gene is, what is that? Isolated and cultured. After that, it introduced into the host virus. The virus is reintroduced into the bone marrow cells. What happened? The bone marrow cells, what happened? Multiplication takes place. The particularly fully functional ADA genes. Okay, so the functional ADA gene produce more means what happened? Automatically the immunity power is restored. That is very, very important. Particularly humoral immunity was restored. That is very, very important. Humoral. Sometimes they may ask the question in this part also. Humoral immunity was restored. If the gene is isolated from the marrow cells, producing ADA is introduced into the cells at early embryonic stage, it could be a permanent cure. Suppose the baby already suffered, Svanti, isn't it? Santi. That girl was identified before the embryo stage, means that particular girl is suffered by a ADA deficiency, like that their immunodeficiency means we have to do this process and introduce the embryonic stage, that it will be permanent cure. After the delivery, what happened? Once we introduce into the embryo in the particular baby gene, isn't it? So it will grow inside the fetus, inside the mother stomach, isn't it? So once it deliver means what happened? The particular baby, it will not suffer by the ADA gene because already we modified in the embryonical level. Okay, it is a permanent cure. After that, what happening? It will he or she will undergo the marriage or some other things. That fetus, it will not affect the ADA diseases. It is a permanent cure that is done by the gene therapy method. Okay, that is very, very important. See that one. So this is the important one. Bacterium carrying plasmid with cloned abnormal ADA genes. Okay, genetically disabled retrovirus. So here we are cloned. After that, this is isolated, cultured, reintroduced in the embryo stage. That baby is not suffered by the ADA deficiency because it is restored. Okay, this is the another important method. And next, that's all about, my dear students. We will pass on to the next one, is question part. Here, first question, see that first, how many recombinant therapeutic have been approved for human use the world over? World over, how many recombinant therapeutic drugs related one, only 1 dozen. So that is denoted 12, 30, 20, 18. This is a number aspects. This is the numerical aspect is there. So 12 only is there. Second one is, which method of cellular defense is common in the all eukaryotic organisms? Already we discussed which method cellular defense. Where there is a cellular difference means it's a RNA interference. Reverse transcript, it is not come. Variable number of trans, it is not comes. And phagocytosis, it will not come. RNA interferences. Don't forget, my dear. Next one is, which of the following technique is used to detect the mutation in gene in suspected cancer patient? Mutation in genes is mutation, nothing but the cancer. Heavily identified PCR polymerase chain reaction. The particular gene is also amplified so many times. Which one is? ELISA analysis of which one, you know? AIDS, blood analysis, it will not come there. Okay. Cancer patient means tissue only, cell only. Page, polyacrylamide gel, electrophoresis. This is also which one, you know? PCR only used for the gene is responsible for PCR only. So PCR polymerase chain reaction only. So then only the isolation of gene is also undergoes the amplification process. Which gene is responsible, like that we can easily identify it. Okay. Go to the next one. The first clinical gene therapy was given in which year? Already you know about 1990. It is not 1991, 2000, 2003, 1990. Don't forget, my dear students. And then last one, I think the clone of sheep dolly has been made by, it is not a gene transfer. Don't forget, my dear, gene transfer is not done by the sheep dolly. Somatic cell, already in our, there are two cells isolated from the different parents, and also the enucleated one, and also inserted the another. These are the somatic cell cloning. Nuclear transfer, its nucleus is not transfer. Germinal cell cloning, it's not comes under this cutting. Okay. And the last one is called as, what is it, you know? That's all about today, my dear students. Okay. So the part, so what is that? 3 will be finished. Okay. So we'll pass on to the next one is part 4. Okay. We'll see the remaining things on the next class, my dears. I'll give you a small in summarize. Okay. Basic gene therapy already know about. Okay. What is the meaning of basic gene therapy? What is my gene therapy? Who did? When? Where? Like that. You have to analyze who is the first gal. Ashanti D. Silva. Which year? 1990. Already know. Basic gene therapy. What is that? All those things already know about. Types of gene therapy. There are 2 types of gene therapy have been reported. One is called a germline gene therapy. Another one is somatic gene therapy. Okay. And that things also already know about. And then how it will be, already we discussed how it will be black. There is a blacking gene is also present in it. So it is also look like an on-off method. And gene therapy can be used at 2 different levels. One is called as embryo therapy and as patient therapy. Embryo therapy, we did, you know, that gene ADA deficiency. And patient therapy is nothing but the bone cancer or whatever it may be, AIDS patient or whatever it may be, like that, already know about. Okay. So in vivo gene therapy and ex vivo gene therapy, already we know about. In vivo gene therapy, ex vivo gene therapy, there are 2 differences there. Ex vivo gene therapy means cultured one, then also introduced into the patient. Okay. So techniques, what are the techniques? They were identification, isolation, construction, transport, integration, and vector also carries. Okay. And also a safe, the vector should be in safe in manner, and also how it will be done. Gene therapy was first applied in the gal, 4 years gal, Ashanti D. Silva, suffering from the immune system deficiency in the year 1990. Okay. Which treatment has given for this particular girl, you know, S-linked server company immunodeficiency, that is called as X-linked SCID. Okay. That is very, very important, my dear. And key ADA deamination enzymes, how it will be, it is responsible for the T cell production. So all those things already know about. With the help of the enzyme is called as ADA, adenosine deaminase. Okay, that's all about, my dear students. We will see the remaining things on the next class. Thank you so much for the participation. Bye, take care, and good night. Thank you.
Thank you, sir.
Thank you, Ajay. Thank you.
Thank you,
ma'am.